Okay, we're live. Good afternoon, everyone. Welcome to Ask the Docs, a Fertility Institute of Hawaii live stream. So this month, we're doing a celebration of World Embryology Day. And so our topic is what is an embryologist and what happens in the laboratory? So thanks for joining us. We have a lot of great information to share. My name is Dr. Carmone. I'm here with Dr. Goulet. And today we have our special guests, senior embryologist Pauline Garrison and Linda Coelho.
Hi, guys.
Welcome, guys. So I think we can start out by maybe one of you guys just talking about what is an embryologist? What do you guys do here?
Yeah. Do you want to go? So the embryologists are the people that hide out in the lab. You hardly see us, but we're there. We're the ones that handle all of the eggs, the sperm, the embryos. We do the biopsies. We're the ones that are loading up the catheters for the transfers. We're doing the inseminations. We're the workhorses.
We're essentially watching them grow until they get to about day 5 or day 6, and we watch them very closely until we can hopefully get a good embryo at the end.
All right. And, I am actually interested. I have this little cheat sheet here, all these questions to ask you. So I don't get stuck in my head, but I'm going to skip around here because I really want to know. Tell me what a typical workday is like for you, and maybe how that's kind of changed since you first started working. How has the field changed altogether?
Yeah, that's probably a good question for you, Pauline, because you have so many years of experience.
It looks different. Well, a lot of things have changed and a lot of things are pretty similar. So I think QC is kind of the backbone of where everything starts. That's checking all the temperatures, making sure the equipment's running smoothly, all the incubators, the microscopes, everything's so temperature dependent, our gases, our CO2 concentrations. So that's kind of where we start. Every morning, we just QC all the equipment, make sure everything's functioning properly, making sure nothing happened overnight. Everything's on an alarm system. So if anything happens, we get calls at night and we come in and deal with it.
So that's
kind of this. Yeah, that's a big part of it. That's kind of everything rides on that. Going forward from there, usually we do the FERT checks in the morning. That's checking the fertilization. You can actually see the fertilization on the, I guess it's an early embryo. So you'll see both pronuclei, one from the egg, one from the sperm. So we do that, we check them, we send the reports out. Usually by that time, we'll have a retrieval starting. So somebody will be starting a retrieval. We'll start doing the thaws for that morning for any frozen embryo transfers for that day. The next person that comes in usually starts doing the checks. So that'll be anything for day 5, day 6. So this is looking at blastocyst growth, seeing if there's anything ready to be frozen or biopsied. And then around that time, people will start doing the biopsies. If the blasts are ready, we'll start doing the transfers. And then usually enough time has passed from the retrievals in the morning to start doing the inseminations 4 hours later. And then that's kind of it. And then towards the end of the day, we just finish up any cryos that we have and start shutting down the lab. But I mean, that's kind of the same from how it was ever ago when I started.
Yeah.
But we do a lot more biopsies than we used to do.
Mm-hmm.
Yeah, when I started, we weren't doing biopsies at all. And then we started doing day 3 biopsies where we were just taking one cell out of an embryo. Kind of realized that's not the way to go.
And of course, technology got so good that we're able to grow our embryos to day 5 and day 6 now. So that's been a big change. We don't have to get them out the incubators on day 3. So that's been a bit of a change.
Yeah, we used to do slow freezing also for embryos. So you would load up all of your eggs or embryos that you're going to freeze, and we would do larger volumes of freezing. Sometimes that machine would take 2 hours to freeze. We can do vitrification very quickly now. So yeah, it's been some changes.
Okay. What about the kind of typical success rates and, how do you see, like, what points in the lab are going to really impact IVF success? I mean, I think it's everything, but maybe there are kind of some key areas that, you know, really only the most experienced embryologists do, and key areas that, you know, are sort of the most impactful for one particular patient's clinical care.
Yeah, good question, and a hard question too. But I think, as you said, absolutely everything along the way. So while we are babysitting the embryos, I think everything that we're doing can impact the quality of it. But I think it sort of starts right at the start with the egg and the sperm.
Yeah.
So if we can have a egg that was retrieved, it was mature egg, we were able to ICSI it with a sperm that had good morphology, I think that is where the sort of the start. And we can sort of, if we have the starting ingredients, then the rest we can work with. When those start off, when egg quality is a little bit poor or when sperm quality is a little bit poor, it doesn't make it impossible, but it makes it a little bit harder. I think that's the most important area. And that's where if we have a good number of eggs, if we have a good number of sperm, I think that's where we can see.
And you'd be surprised how, we were talking about the technology changing, you'd be surprised how much we come back to the basics, which is what was, you know, if we have a case and we're kind of wondering, oh, there's not as many blasts as we hoped on this case, you know, the eggs did look good. And, you know, we just go back to, was the temperature okay? What was the pH like in this incubator? We really go back to just the basics because it's really kind of the housing where those embryos are growing. That's kind of where they're going to be most of the time. So, you know, as much as there's bells and whistles of biopsying and PG, you know, everything else, it's kind of, you know, the temperature comes back to it. We still talk about media. Are we optimizing the media? Are we optimizing the culture system? So there's a lot of factors, but you'd be surprised how the basic stuff, you can't look past it.
And that's important.
Right. Yeah, I mean, even air quality, and things like, you know, the type of paint on the walls and the, you know, everything seems, even the tiniest little things seem so relevant and have been studied. And that's why, you know, for patients who might be watching this, if you guys are wondering why we tell you, you know, not to wear perfumes on your day, because all that stuff can impact embryos in ways that obviously we can't necessarily measure once the embryo is back inside you. But yeah, that's where it's not because we don't want to smell the perfume. It's
the embryos don't want to
smell it. Right. Right.
But you know, I have patients who are worried about their embryos and the care of a babysitter that they have never met before, and they want to know if we're keeping a close eye on them, and they ask if we get them out and check them every day and how's the development going. And I tell them, I reassure them, you don't want us getting those embryos out every single day and checking on them, because that means taking them out of their cozy incubator, exposing them to cooler temperatures, exposing them to the outside environment. Just like a fetus belongs inside the belly, those embryos belong inside the incubators. And we even have you know, state-of-the-art top technology. We even have that embryoscope that will watch our embryos and take time-lapse photography of how our embryos are doing. So between all of the alarms and bells and whistles and camera videos on these embryos, we're able to get a lot of information about how these embryos are doing, and that you guys can assess and monitor and help determine which is the best embryo to recommend.
Yeah, and that's a good point too. Anytime we're doing anything in the lab, you try to think about, do I need to be doing this right now? Is this an unnecessary step of taking things out? Do I need to do 2 checks for this one thing? Or maybe I can just put this all in 1 check. So we are very mindful of how many times we're taking things out and putting them back. And like you said, the changes, you know, in this tiny micro environment, you know, anything we can do to cut that out and just make it stable for the embryos, the better.
They're pretty good at growing. So the less we can interfere with that growing process, they like the dark, the warm incubators that are gassed at the perfect CO2 and oxygen levels. So
they don't really
the
need
more
us.
we interfere, the less we interfere, the better. So
You bring up a good point, that embryos are good at growing, but we know we don't expect all of the embryos to make it from egg stage to fertilization and then to day 3 or day 5. What is your take on that growth process and the attrition rate with that?
Yeah, so there is that attrition rate, and there is a drop at almost every little stage of the embryo's little lab time. So first of all, we have a first drop off that isn't, well, it is sort of lab related. So we have the number of eggs collected. And so the first drop off we see there is we can only inseminate the eggs that are mature. So we do expect a good cohort of them to be mature, but as you are more than well aware, that some cases we have a good maturity rate and some not so well. So that's our first sort of drop off there. And then they fertilize, and we have great fertilization rates.
Yep.
With ICSI, we're seeing upwards of 90.
Yep.
90% of those eggs fertilized. So then that's another drop off. So if we started off at 10 eggs that were mature, now we're working with maybe a 9 or 8 to 9. And then from then they grow to day 3. And at day 3, another sort of cellular process happens in the embryo, which is another stage that we generally see that we see embryos drop off. And the checkpoint there is actually really interesting because it's when the maternal DNA stops operating and helping out, and it now becomes the embryo's DNA, and it switches on and it has to continue for the growth of the embryo. So we see another drop off there. Again, not too many. We expect that about 80% of those embryos get to day 3. But then we have to get a good quality blastocyst on day 5 or day 6 or day 7. So out of those embryos that make it to day 5, again, we hope that majority of them make it, that it is another drop off. Number one reason why embryos drop off is usually, as I said, way back in the sort of egg stage, that egg has something going on at a genetic level, even the sperm. So way at the start is sort of impacting what we see on day 5 and day 6 and things like that. But we can aid that process with, like you said, making sure all our equipment is all right, that we are interfering as less as possible, that when we are working with the embryos, they're getting right back to the incubators as soon as they can. So
you bring up an interesting point right at the very beginning, and I'm going off.
No, please.
Not according to the questions that we have pre-written up here, but the maturity of those eggs. You know, we hope that the majority of those eggs that we get out are mature. One of the most frustrating things that can happen is when they're not mature. And a common question I get is, well, can't you continue to culture them and force maturity on them? Can you just like a kiddo, can you give them time and allow them to grow more? And what's the current state of research and ableness for us to do that right now? Ability for us to do that?
Yeah. So eggs, it is possible for an egg to mature outside the body. So we can have an immature egg that is retrieved at an egg collection, and it is very possible for it to mature later outside the body. And I think what you're referring to is in vivo maturation, so IVM. And it's where some clinics have been looking into research about adding not artificial, but they add
specialized
media to aid that egg to mature. What we do know after years of research is that those eggs, unfortunately, aren't always going to have the best fertilization, not compared to the eggs that were mature at retrieval. So we know that the eggs that have the best chances are the ones that were mature and didn't mature later. We know that.
We do have those cases in the lab where if somebody has a lower than projected maturity and the egg yield is quite low, we will offer that for the next day. We will hold on to them and try to inject them on the next day. The fertilization tends to be lower. The blast development tends to be lower. We do get blasts out of there, and we do get normal biopsied embryos out of there too. It's just going to be at a lower rate. So we don't do it routinely, but we do it for the low prognosis patients, the low egg yield patients, or the patients that had a lot retrieved but only have 1 or 2 mature. We will hold on to those and try that, because at that point, I mean, it's worth it for the patient to get a shot. So,
and I find that very reassuring that you guys really do go the extra mile for our patients to try and make every egg work and make it into an embryo that can lead to a pregnancy.
Yeah. And I think the process of in vitro maturation, or IVM, right now is still in pretty early stages. So that's still something that the
yes,
people
very
who research on it are kind of still trying to figure out. But that may be possible. I mean, the idea is, wouldn't it be nice too to be able to get eggs that, you know, without going through a stimulation,
Yes,
you know,
right.
or minimal stimulation, get eggs out and mature them along the way, or something like that.
Yeah, I can see why that's appealing. Imagine that.
But that's also part of the idea with kind of in vitro maturation, which is something we're not really doing right now. But
Yes.
but the other thing I wanted to point out about that was that, you know, the natural attrition that you guys talked about really is part of natural human reproduction. And, you know, overall numbers suggest that maybe only 70% of eggs, 7 or 8%, not 78%, percent of eggs really are able to yield live births. And so that number sounds daunting, but that's exactly why we try to mature as many eggs as we can safely during IVF, because we know that there is just a natural attrition process. And even with donor egg cycles, young donors who don't have any known fertility problems, who are in their 20s, not all of their eggs are going to fertilize. Not all those embryos are going to yield blasts. Not all of them are going to grow to that final stage. Not all of them are really meant to be, because there are so many hoops that that these little embryos need to jump through. And, you know, that's why natural human fecundity, natural human fertility, is not nearly as good as I don't know, I would want it to be, I suppose. But ultimately, even if you have no fertility issues at all, you're in your 20s and you have unprotected sex in one particular month, what are the chances that you get pregnant? It's really only around 20% or so.
Yes.
And that's probably because not all those little eggs are getting fertilized. You know, there's only one egg, and may or may not get fertilized, and may or may not continue to grow and implant and all that. And so that's very different than the human, you know, the reproduction of bunnies or something like that, where, you know, they get completely. Great. I have bunnies, but they're both male. They're not getting pregnant. Maybe someday. So I think that it's important to keep in mind, I guess, the expectations that we need to set as physicians for our patients, that not all of these embryos, not all the eggs are meant to be. And that's why we try to collect, you know, 20 eggs, not for you to have 20 babies, but for you to have, you know, a small number of babies from those 20 eggs.
That's a good point too, because even for us, I don't know, on the lab side, it's you go, you know, you process so many embryos in a day. And I mean, you can be sitting there and doing a transfer on this beautiful blastocyst, and you know it's PGT normal, and the patient can still not be pregnant, or vice versa. You're transferring this poor little embryo that doesn't look like it's going to make it, and
it turns out to be healthy.
So it's a very humbling field, even as, you know, as long as Combined we've been doing it. It's still, you think you know something and you're always proven wrong. I mean, and it's not even in a bad way. It's just, it makes you realize you really don't know as much as you think you know. There's still so many things that are out of our hands, and a lot of things we don't even really understand how the processes work to get people pregnant. So there's a lot of things there to think about, even for us. I mean, we all sit here and we think we know what we're doing, but a lot of it is, I don't know why it worked, or I don't know why it didn't work, you know?
And at the end of the day, human biology, there's so many. You can say that eggs are supposed to look like this and sperm is supposed to look like this, but with just like humans, there's so many variations, and some might be just slightly different and every egg is different, even though we can't see it. Sometimes we can see it, but every egg, every sperm is different, and we don't fully understand quite yet.
You bring up something I get asked a lot, is how do my eggs look after my retrieval, or how do my embryos look? How do you evaluate that this was a good egg, or how do you evaluate that this was a good embryo? Aside from the genetic testing that we can do on the embryos, what are you looking at under the microscope?
Yeah, so we're talking, we refer to it as a morphology, or how they appear. And we can Google what an egg looks like. You can Google what a sperm looks like. There is a bit to it, but generally what we're looking for are the structures and signs to us that sort of tell us that it's a healthy egg or a healthy embryo. So we do grade our embryos with eggs. It's a bit different because they're one cell. So we're sort of looking at one cell. At retrieval, when we're collecting them, they look very different from after we wash them and we clean up all the cells. But after all the cells have been removed and we're preparing it for ICSI, that's when we can tell it's mature. So first of all, we can look at if it's a mature egg, and if it's, when we say check maturity, we're looking for a little polar body on the side of the egg. So you'll most likely see a cell with a little tiny cell on the outside in the shell. That tells us that it's a mature egg. So first of all, that's one of the things we're looking at. And then we can dive a bit deeper if we want. We can look to see what's the consistency of the cytoplasm, which is sort of the cell in the middle. Sometimes they appear grainy, sometimes they're really plump.
And that ends up kind of being the person, whoever's doing the ICSI, is sort of the person, you're almost feeling it with the scope as you're doing the injection. So you kind of have a better feel of what the eggs popped like, how much that ooplasm gave you some resistance. Did it pop too easily? Did it pop too hard? You can kind of feel, and it's hard to explain because you're using a micro needle inside. You can kind of feel the consistency of the cytoplasm. Sometimes it feels gummier, sometimes it's a little too loose. And we'll write that on the report and just say, oh, this felt a little off, you know, and give feedback. So that's kind of how we do the grading. I mean, we can look at the cytoplasm and look and see, like Linda said, about how smooth it looks, whether it's grainy, that kind of stuff kind of gives us some insight.
So only really after you've done it a whole lot of times, after you've performed lots of ICSI, then I think you can say this egg felt a little bit structurally, it was structurally not really there, whereas this egg had really good resistance. It accepted the sperm really well during the ICSI procedure. So after we sort of do these procedures, we get an idea of how the eggs work. But as we said, those eggs that we think, oh gosh, they were a bit dark, end up being beautiful blastocysts, and other eggs that we thought were beautiful can, you know, not fertilize. So as I said, it's the nature of biology that we can assume and we can guess and make our notes, but it's all a numbers game at the end.
Yeah.
I usually also tell patients that with eggs, we're not really, there's not a kind of universal grading system. I mean, sort of what you guys are talking about is getting a feel for what these eggs look like based on your experience, but it's not quite like with, you know, blast embryos where they're given sort of an actual grade that this can be sent to other clinics and the clinic then understands that.
Yeah.
And so usually I tell patients that we're just, you know, we're just going to see how those eggs perform. That's the test. The test is how are they gonna do when we put the sperm in there and try to make a baby. And even with grading of embryos, just a side note, it's tricky because if the embryos are sort of fair to good quality, they don't predict live birth very well.
Yeah. And that's what I think we like to remember.
Yeah, I mean, we need something to be able to discriminate between embryos when we have a bunch of them. But I definitely tell my patients, I communicate to them that they really don't need to focus too much on the grading. I mean, it is what it is, and it is not reason to fret as long as we have, you know, sort of decent, freezable quality embryos that we're able to transfer. Success rates are not all that different. Between the different grades, and then especially when we're looking at PGT tests and embryos, I'm so surprised that it's that perfect A quality embryo that is abnormal, and then that, you know, whatever BC that is we thought wasn't quite as good, which is fine. And PGT normal embryos, again, as long as they're decent quality, they have a 70% success rate, something like that, you know, regardless of what their grade is. So it's, you know, sort of not as important as I think it once was thought to be.
Yeah. And that's a really good point. I'm glad you bring that up with the patients, because internally we kind of have a checkpoint system within the embryology group, and we will put embryo up on the scope and say, what would you grade that? And just kind of make sure we're all kind of grading the same, because it can be pretty subjective. And sometimes it's the angle of the embryo. Remember, these things are 3D. So you all of a sudden turn it a different way. And now all of a sudden it looks more like an AB versus a BC, you know, or something. So there's a lot to go into the grading. So I'm glad that you bring up to the patients, don't put so much emphasis on that, because then also after freezing and thawing, it could look so much different. And then they usually are in the incubator a few hours before the FET. So a lot of things are changing and they don't always look the same before the freezing and at the FET. So,
and what I like to say, because it's easy to get caught up on grades and how many AAs and how many BBs I have, but what I like to say is it's a tool. Grading is a tool that the embryologists use to describe what they're looking at, and that's really all we can do. We're not looking at the genes. It doesn't mean an AA is a healthy embryo. Likewise, it doesn't mean the CCs are abnormal. It's a tool that we use to describe. Pauline, did you see that 4AA that we're working with? It's a tool we use.
And I already have a picture in my head of what I'm going to look at when she puts it up. But it's not necessarily this is going to be that much better than a BC. You know, I mean, it's going to look better, but who knows what it's going to biopsy like? Who knows what it's going to freeze and thaw like?
Earlier, you both had mentioned that after you do a high number of the ICSIs, a high number of the injections of the sperm into the eggs, you start to develop a feel for what the egg is supposed to feel like, or for how the sperm is supposed to behave. And I think that's so important, because I know a lot of my patients, sometimes they're a little overwhelmed by the whole process of everything, the costs of everything, and they're worried about the babysitters that they've never met before taking care of their eggs and sperm. And I assure them that you guys are very highly trained, technically skilled. You are the first surgeons that are operating on these embryos, and not just anybody can do this. Can you talk a little bit about what's involved in becoming an embryologist?
Yeah, definitely.
So we come from kind of all walks. We, at some point, you know, a lot of us have master's degree. We have a PhD in our group too. So people have the academic background plus the hands-on training. So everybody that comes into the lab before you get started on anything, you know, even if you come from another clinic, like Linda came from Australia, we have Arina that came from Japan. You know, we're never just going to let you walk in and just start doing patient samples. We get everybody in line. There's a strict training. You know, we make sure that your rates are well enough before you can do patient samples. So we try to get everybody in line. And like you, I think we were talking about rates before and ICSI and what the fertilization, we try to make sure we're all within the same percentage points of ICSI rates, transfer rates. So, you know, there's nobody on the outliers here, but yeah, everybody's trained up. We all participate in proficiency testing, so everybody's on the same page. We do quarterly analysis of our rates, so we're all in line with each other for sure.
I think there's a lot of hands-on training. It's pretty impressive, and then people need to be sort of signed off before they can proceed on to the more advanced stage.
It takes years.
Yeah, for sure. Before I forget, a very, very important question for our patients. Can you guys go through what are the kind of precautions and measures that you guys take to ensure the safety and security of the lab, to ensure that we are transferring the embryo that we say that we are transferring, to sort of ensure that we are correctly identifying eggs, sperm, embryos. Can you take us through that stuff?
Yeah, so what's really interesting is, yes, we trained so thoroughly on ICSI and on biopsies, but another thing that is taken it is taken more seriously than all that is our witnessing procedures, our IDing, our labeling. So if anyone's been into any medical procedure in an IVF clinic, you get asked about 3 times, what's your name and date of birth? And that happens with every embryo dish in all our labs. Every embryo dish, every label, we are checking multiple times. So
every time we move from one dish to another dish, every time we're transferring things, if you're doing a thaw, there's always somebody else right there with you, double checking the names, dates of birth, numbers of embryos. So,
and I think that's important because it doesn't matter, you know, how much experience you have or what your degree is in. Someone's going to check that your check was correct. So, we do double check each other's work. We check it. We get another set of eyes.
Yeah.
And everything is so, well, it has to be so stringent.
Yeah, and we kind of, I mean, we trust each other we have to. We work so closely together. We're such a tight group. But at the same time, we tell each other, don't trust that I didn't make a mistake. Don't think that I, you know, when you're checking me, look at those dishes as if there was a mistake there and it's your job to catch it. So we're pretty tight on, you know, making sure just because, you know, Linda's doing an ICSI, don't think she's got it down, you know, just make sure you're there, read all, and, you know, we pick up on things, this little date of birth, even if it's off by a year.
One digit, that is.
One digit, we pick it up, you know. So yeah, we're pretty tight in there on those double ID checks.
Sure. There's a question from our audience here, but I want to finish this topic first. There are also alarms, and there are also right. Yep. So there's alarms, and then there are locks. Can you guys just mention where those checkpoints are?
Yeah, definitely.
So the whole lab is on lockdown
all the time from everyone but us.
The lab is locked up. All of the cryo rooms, which is where all of the tanks are, get locked up.
And those are separate locks than the rest of the
locks.
So
yeah,
typical. So clinic staff we have just access the labs. Yeah.
Right. So all the doors to the lab get alarmed at night. The cryo rooms are alarmed at night. All of the equipment is alarmed. So all of our gases are getting checked through the night. Temperature is getting checked through the night. Those tanks are also set on alarm. So if any of the liquid nitrogens should happen, the levels should go down. We'll get calls. We have backups for everything.
Absolutely.
If an incubator goes down, we have a different incubator we can move things in. We have an entire completely empty liquid nitrogen tank that we can move things into if we needed to.
And what Pauline said was really cool is they're all linked to an alarm system that calls us. So all our mobile numbers.
What's really cool is we get called at
night if it was to happen. So everything, even though it alarms, it's alerting multiple people at that time. So it's not like we have to wait till the next day to come in. We're getting those alarms straight away.
Okay, and then we have a couple of questions here just before we wrap up. So we have somebody asking, how are embryos graded? What's the difference between grading versus PGT testing, and which is more reliable? So I think we can answer some of those, but maybe you guys can answer, how are embryos graded?
So it's a grading system that is based on the expansion. So basically how big that blast got. Mm-hmm. And then there's two letters so the expansion is the number. It's 1 through 6. And then we have, we do 2, right? 2 through 6. Mm-hmm. We don't use the 1, but then there's 2 letters that go along with it. And the first one is the inner cell mass, how the quality of it, how big it is, how tightly it's compact. And the second one is the trophectoderm. So it's sort of the mass of the cells that becomes the baby. And then the trophectoderm are the cells that would become the placenta. And then you have your zona. So that's sort of keeping the whole embryo together. Yeah. So if you have a 4AA, for example, is a well-expanded blast with a really nice inner cell mass and a nice trophectoderm. And then B would be a little bit lower. And then a C grade would be a little bit lower than that.
So it's basically just kind of a description of what the embryo looks like physically, mm-hmm. right? Yeah. And it's all done under the microscope. And again, there's some subjectivity there a little bit, right? As we were talking about before, embryos are 3-dimensional, 2D, right? They can look, and it's a dynamic process. Embryos grow, and so blasts can expand, and then they can shrink, and right. So they can look different at different times.
Right.
So that can influence that process.
Checking on something on day 5, first thing in the morning, versus looking at them in the evening when you're closing down the lab, that same embryo can look very different.
Totally different. Absolutely.
Compare 2 day 5s to each other, or even a day 5 or a day 6.
And then what's the difference between grading versus PGT testing was part of that question. So PGT testing is where we actually biopsy. We take a small sample of the outer cell layer of the trophectoderm, which is the outer layer of the embryo. That's what differentiates into the placenta. That's what sort of implants within the uterus. We take a sample from there. The embryologists take a sample from there. That gets sent to a specialized genetics lab to evaluate the genetics specifically and look for things like chromosomal disorders, mainly looking for things like Down syndrome. Okay, so you can have a perfect 4AA embryo, and you can do that genetic testing, and that embryo can have an extra chromosome or be missing a chromosome or pieces of chromosomes and things like that. Extra chromosome 21 is Down syndrome. That's kind of the famous one. That's why I'm using that as an example. But there are certainly many, many, many disorders or issues that an embryo can have. So there are 2 totally different types of tests. Which is more reliable, physical grading versus PGT testing? Well, the physical grading is always done, right? Because, we have the embryo in front of us. We're always going to grade it. It's there. We're looking at it and we grade it. PGT testing. So if you're asking what is more reliable to predict live birth, it is going to be PGT testing because, the grading of embryos can be utilized sometimes to predict whether it will be a PGT normal embryo, but it's the PGT testing that has a much stronger association with live birth. You have to understand, though, that any of these tests do not make an embryo normal or good. These are just, they just give us that information ahead of time. So, you're welcome. Patients are thinking. So, many times patients will perseverate over, is this a good embryo? And certainly I can understand that from a patient perspective. But on the other hand, the only way to really know is to just put it in there and see what happens. Because ultimately I can give you probabilities based on literature, but we don't really know until we try.
Right.
So. Were there any other kind of important points before we wrap up our session here? Because I think we touched on a number of different things, right?
We had some good questions.
Good. Well, thanks everyone for joining us today. This is Ask the Docs, the Fertility Institute of Hawaii live stream. For more information, please check out our website, ivfcenterhawaii.com. If you have additional questions, you can leave comments below here, and we'll kind of answer them later on. And of course, you can always reach out to us, schedule a consultation with us. If you're already a patient, then you can always just ask a quick question through Clara. Otherwise, we're excited to meet you. All right, guys. See you soon.
Bye.
Thanks for having us. Thank you.
Bye.